Hello, welcome to the next in the series of review clips for your Biology Theory 201 public examination. In this clip, we'll be taking a look at the chromosomal theory of inheritance and how it came to be that inheritance patterns was viewed, determined, discovered to be linked to chromosomal inheritance patterns. The real answer is that chromosomes seem to follow the same behavior as Mendel's factors of inheritance. But this alone didn't prove that chromosomes carry the genetic material for inherited traits. The proof for the chromosomal theory came from experiments done by Thomas Hunt Morgan and Calvin Bridges on the simple fruit fly, Drosophila melanogaster. Before we move forward, again, we need to be familiar with a few definitions first. Karyotype is the number, form, and types of chromosomes in a cell. Heteromorphic chromosomes are chromosomes that act as a homologous pair but are different. Sex chromosomes are a type of heteromorphic chromosome that determines the sex of the organism. Autosomes are all other chromosomes besides the sex chromosomes. The Y chromosome is the sex chromosome that only occurs in males. The X chromosome are sex chromosomes that occur in both males and females. Sex-linked is any gene that lies on the X or Y chromosomes. Wild type is a term that geneticists use for the standard or reference type, the type that is quote-unquote normally present. And mutant is anything that is different from wild type. What you see in front of you here now is a normal karyotype for a human male. Chromosomes are linked, paired according to a banding pattern and size. And in a male, a normal male, you would have 23 of these. 23 autosomes, which I'm circling here, putting a square around here, for a total of 44, plus an additional pair, XY, for the sex chromosomes. It is the XY that determines the sex of the individual. Here you see it again by direct comparison. The difference between the two is that in a male, you see XY, or XX rather in this case, whereas in a male, you see XY. Thomas Hunt Morgan did a number of experiments that actually looked at and listed and actually gave credence to the idea that the chromosomes were indeed the basis by which heredity was passed on from generation to generation. Initially, Morgan was certainly skeptical about Mendelianism and chromosome theory, but ironically enough, his experiments provided strong evidence for both. His experiments done on the common fruit fly. Why did he choose the fruit fly? For a number of reasons. Firstly, they were small, they're easy stored in vials, and very simple food source. They're easy to keep, easy to maintain. Secondly, a single mating would produce literally hundreds of offspring, so you had a lot of data collected from a single breeding. Third, they have a short generation time, 2 weeks. And as he was doing experimentation on the chromosomal patterns, the fact that they have only 4 pairs made the analysis much simpler. In fruit flies, eye color exists in a number of forms. Wild type is, as you see in front of you, red eyes. The white eyes is a mutation. So in 1910, Morgan received the Nobel Prize for his work, which was awarded to him in 1933. His experiments with fruit fly demonstrated the chromosomal theory of heredity. He discovered a mutant white-eyed male where the wild-type color is red. He then crossed the wild-type female with a white-eyed male. All the F1 had red eyes, white is recessive. When he crossed the F1 generation to get his F2, he found he did get that characteristic 3-to-1 ratio. But the significant part that he noticed was that while he did get a 3-to-1 ratio, all of his white-eyed flies also were male. This led him to hypothesize that eye color gene is indeed located on the X chromosome. And he did see a ratio was actually of 1 to 4.4, which is less than 1 to 3, but white-eyed flies have a lower viability. They don't survive in as large numbers. So Morgan made this white-eyed fly mutant with wild-type flies. He got F1 all red eyes, F2 to 3-to-1 ratio, red to white. But, significantly, white color only in males. All of the F2 females had red eyes, and with the males, half had red eyes, half had white. And here you see the eye color illustrated again, red versus white. So, what Morgan wondered was that, is it possible to get a white-eyed female? Under what circumstances? Looking back at the cross, if you take the red-eyed female with a white-eyed male, he hypothesized that the gene for eye color was only found on the X chromosome. The eggs that the original female, the red-eyed female, would produce, wild-type female, was all wild-type. As for the male, he would produce an X containing the white-eyed gene and a sperm which contained no gene. When they were crossed, the yield was females that were, for all intents and purposes, heterozygous, and males that contained a single gene, one for wild type on their X, and the sperm on the Y, which made them, of course, male. When he crossed the F1s, the resulting female could produce an X with a wild, an X with a white. The male would produce an X with a wild and a sperm with neither. The result would be a red-eyed female, a red-eyed female, a white-eyed male if he received his X from mom with the white-eyed gene, and a red-eyed male. The ratio was indeed 3-to-1, with 3 wild type, 1, 2, 3, but there was indeed a single white-eyed male for an overall ratio of 3-to-1. To get a white-eyed female, the cross would have to be as follows. Remember, a white-eyed female would be X little r, X little r, where big R represents red, little r white. In order for that to happen, mom, the female fly, must have passed on an X little r. as would the male fly. So in order for a female to be white-eyed, the male parent must be white-eyed. The female fruit fly could be red-eyed, providing that she is at least a carrier for white eyes. But the male would have to be white-eyed. Of course, to complete the square, and this cross would yield one wild-type female, one wild-type male, one red-eyed, or white-eyed rather, female, one white-eyed male. But in order for a female fly to be white-eyed, the male fly, the male parent, must be white-eyed, and the female fly must be at least a carrier for white eyes. So to recap, when the white male was crossed to wild-type females, all the progeny were red-eyed, white was recessive. When you cross the F1, you got 3-to-1 ratio, but the white was only seen in males. Morgan found the white-eyed male while looking for alternate alleles of genes. Single male with white eyes that showed sex linkage. And he did experience with them. Crossed red female, white male. All the progeny were red-eyed. This can be explained by looking at the following cross, where the crosses were only found, the genes were only found on the X chromosome and not on the Y. So we did the parental cross, red eyes, white eyes, and possible gametes being produced are as such. When you cross them to create your F1 generation, you get the following. All red eyes. But note that the female is indeed a carrier. When you cross your F1, the possible gametes are as you see here. And the F1 cross yields 3 red eyes, 1 white eyes. So when you're talking about sex linkage, again, this is one thing. So the most significant work that Morgan did was actually draw this analogy, draw this conclusion that traits were indeed carried on the sex chromosomes. If you look at, there are some things you need to keep in mind. Firstly, Y chromosome are only transmitted from father to son. A father's X can only be transmitted to a daughter. A mother's X chromosome can be transmitted to either son or daughter. Males get their X from mom and their Y from dad. And if you're talking about a trait, an allele that lies on X or Y chromosomes, they will follow the above rules. When we are looking at genes located on sex chromosomes, these are sex-linked genes. And doing a reciprocal cross can indeed reveal sex linkage. So we look at chromosomes and Mendelians. These two are linked. The reality is that cytology, cell biology, and genetics came together at the beginning of the 20th century. We know the chromosome theory of inheritance. Mendelian genes have specific loci on chromosomes. and it is the chromosomes that undergo segregation and independent assortment. The two are tied together. So Morgan helped restate Mendel's law of independent assortment. And we can restate Mendel's law as follows. If crossing over does not occur, genes that are located on the same chromosome will be inherited together, while those on separate chromosomes will assort independently. So with regard to sex-linked traits are traits that are carried on the sex chromosomes, X or Y. Most often, the traits are carried on the X. Most of these traits are recessive, and often they are lethal. And sex-linked traits affect males more often than females. Some examples of sex-linked traits in humans: red-green color blindness is sex-linked. Male pattern baldness, hemophilia, and Duchenne's muscular dystrophy are all examples of sex-linked traits that exist in humans. All of these traits are more common in males. The reason being is that for a male to have a sex-linked trait, they merely need to inherit a single allele. Whereas for females need to have two copies to show the trait. Let's take a look at this cross. A woman who has normal vision marries a man that is colorblind. What are the possible ratios from their offspring? As this is a sex-linked trait, we have to use X and Y chromosome across. Now, making the assumption that the female is homozygous for normal vision, we write the female as X big N, X big N. The male will be X little n, Y. If this was indeed the case, the alleles from mom would be X big N or X big N. And again, note that we attached the gene, the allele, to the actual sex chromosome, because it is indeed sex-linked. For the male, X little n or Y. The cross then yields the following. All the females are carriers, but they are normal. All the males will indeed be normal. The reason being is that the males get their X from mom, and if all of mom's X's were X big N, all males will indeed be normal. Females, on the other hand, because they get an X from dad, would be colorblind. So to keep some things to keep in mind. Firstly, sex-linked traits are recessive, by and large. They're carried on the X chromosome normally, and if a person has a big allele and a small allele, in other words, X big N, X little n, they're considered to be a carrier. For a sex-linked trait that is carried on the X, males cannot be carriers. They either have it or they do not. Females can indeed be carriers if they are heterozygous. In past public examinations, questions have been posed on this topic. For example, what is one of Morgan's major contributions to the field of genetics? Codons. This wasn't Morgan. The double helix, that came later, but again, it wasn't Morgan. The law of dominance, that falls back to Mendel. The correct answer is D, sex-linked traits. Constructive response questions have also been posed, which involve the uses of sex linkage in completing the cross. In this question, you are told that hemophilia is a sex-linked trait where normal clotting is dominant and hemophilia is recessive. Tongue rolling is dominant to non-rolling. So those are your two traits. So, normal clotting, H. Little h, hemophilia. Tongue rolling, but this is sex-linked. Tongue rolling, big T. Is dominant over non-tongue rolling, little t. So with that being realized, we now start working through the cross. Our first step are what are the genotypes of the parents. The genotypes of the parents are as follows. For the male, he is non-hemophiliac, therefore normal. Because hemophilia is only carried in the X, his genotype is X, little h, Y. For the female, you were told that she and the male are heterozygous for tongue rolling, but the female is a hemophiliac. So she is X, little h, X, little h. They're both heterozygous for tongue rolling. Step 2. What are the possible gametes? For the male, he can produce X, big H, big T, X, big H, little t. Y, big T, Y, little t. For the female, she can produce X, little h, big T, X, little h, little t. The other two gametes are simply repeats, so I won't write them in. To do the Punnett Square for this cross, for the male, across the top, his gametes are X, big H, big T, X, big H, little t. Y, big T, Y, little t. For the female, X, little h, big T, X, little h, little t. Completing the square, which I'll do here now. So when you look at the cross, there are 8 possibilities of genetic combinations in the offspring. You are looking for those that are hemophiliac and also tongue rollers. Going through the list, this first individual here, X big H, X little h, is a non-hemophiliac. X big H, X little h, again, non-hemophiliac. X big H, X little h, non-hemophiliac. X big H, X little h, non-hemophiliac. None of our females can be hemophiliacs, as their father, his X, is going to be X big H. The males, however, are all hemophiliac. You're interested, however, in those males that are tongue rollers. A single big T would yield the tongue rolling condition. 1, 2, 3. 3 of the 8 offspring are indeed hemophiliac and tongue rolling. So the answer would be 3 over 8, which equals the percentage that you're looking for. And that percentage is 37.5%. This sample question is posed. Why are males affected more often than females with respect to sex-linked disorders? Females do not carry the Y chromosome and therefore cannot inherit. If this was related to a Y-linked chromosome, yes, indeed, this would be the case. However, sex-linked are typically carried on the X. Males receive this recessive trait on both the X and Y chromosome. Again, this would be relative to whether it was actually carried on one or the other. The recessive trait is found only on the Y chromosome. Again, if that was the case, it's absolutely true. However, sex-linked traits are typically carried on the X. The correct answer is B. They only need to inherit one recessive trait. Therefore, the presence of a single allele being passed to them would ensure that they would indeed display that recessive sex-linked disorder. Another constructive response question is presented as you see here. The question reads, a couple has a son with muscular dystrophy and 2 daughters, one with the condition and one without. All 3 children have a widow's peak. Neither parent has a widow's peak. Muscular dystrophy is recessive sex-linked. Widow's peak is an autosomal recessive. You are interested in finding out the genotype of both parents. So, looking at the traits. Firstly, the children have widow's peak. So if we were to draw out the traits, and we'll do a 4x4 Punnett Square here very quickly. Four by 4 square. We are dealing with a sex-linked trait. So let's write in our male summary here what we know about that male son. couple has a son with muscular dystrophy, a sex-linked trait. So to show a sex-linked trait, we'll write the male up top, female down the side. The male has muscular dystrophy. So to be muscular dystrophy, that child must have gotten, we'll use little n to represent the presence of it, it's recessive, capital N to represent normal. So the male must have gotten muscular dystrophy from mom. Now, all three children have widow's peak. Widow's peak is a recessive trait. So we'll write big S to represent straight, just to keep our conventions, hairline, and little s to represent widow's peak. So he must have gotten a little s from mom and a little s from dad. So what we know thus far is that the mother passed on an X little n. She doesn't have muscular dystrophy. So we know that mom is X big N, X little n. She doesn't have a widow's peak. So she would be big S, little s. That's mom. As for dad, what do you know about dad? Again, we know that he doesn't have muscular dystrophy. So he's going to be X big N Y. He will also be heterozygous for widow's peak, because he doesn't have it, but one of his children did. So dad is X big N Y. And again, on your public examination, very clearly draw a square around your answer to clearly let the marker know what your choices are. Thank you for your time. Thank you for your attention. I hope you found this learning clip to be helpful.